Hi, I'm Dr. Brian Kaufman. I'm a family doctor with CLL, and I'm at ASH 2014 in San Francisco. Dr. Sharman, you want to introduce yourself?
Hi, my name is Jeff Sharman. I'm a hematologist-oncologist. I have a private practice in Eugene, Oregon, but I serve as the medical director of hematology research for U.S. Oncology.
And you've done some really important research, but I'm going to take a big step back because some of that research that you've done and your colleagues have done have kind of, and I'm stealing your phrase, kind of cracked the biology in CLL. So maybe can we take one giant step back and talk about exactly what we know about CLL, what it's doing in the blood, because it's an entirely different understanding than we had even just a few years ago.
So, yeah, no, I totally agree. In a lot of different cancers, we talk about onco-addiction. That's a term that's out there among scientists. And onco-addiction is this idea that there's some specific mutation in the cancer that drives the cancer to grow. And we've taken advantage of that in a number of cancers. You know, melanoma, we can give BRAF inhibitors or other drugs.
If they're BRAF positive.
Yeah, if they're BRAF positive. And
right.
so there's this concept of onco-addiction. Chronic lymphocytic leukemia does not have the same biology of some activating mutation that drives the biology. But there are a couple pathways that are overactive, and we can interfere with that almost in the same way that we use tamoxifen for breast cancer. You know, a estrogen blocker in breast cancer, we have these blockers. And
or androgen blockers in prostate cancer.
Brought in prostate cancer, exactly. The key pathway that everybody's aware of is the B-cell receptor. So let's kind of unpack this for a little bit. You make antibodies to fight flu, E. coli, salmonella, all these things, right? Every B cell has...
Not as well as you do because I have CLL, but I try, yeah.
Hypothetically,
right?
Right? So every B cell has a B cell receptor. And when we give a B cell receptor inhibitor, such as ibrutinib or idelalisib, we see these incredible responses. And so it's clear that this B cell is really important to... This B cell receptor is really important to the survival of B cells, and here's how it works. We don't know exactly where CLL starts in terms of the evolution of the B cell, but B cells start from really early on in the bone marrow. If those go wrong, that's acute leukemia. Then they evolve a little bit further, and that's, you get a cancer from that, it's lymphoma. Then a little bit further, that's CLL. If you could keep going, that becomes multiple myeloma. So it's like every stage of B cell development has a cancer that is associated with it. In CLL, they kind of get locked at this one step where they don't like keep evolving, and they don't regress. In this place where it's locked, the B cell receptor is central to it. So what happens is you have the lymph nodes and the bone marrow, which is like where these cells go to grow.
And to by grow you mean proliferate?
Proliferate, yes, reproduce. Yeah. So they get into the lymph nodes, they get into the bone marrow, and this is where the B cell receptor gets engaged and creates survival signals and growth signals. And so these B cells grow and grow, and they get totally exhausted, and they leave the lymph nodes and go to the blood to recover. And so for the longest time, we always...
So I got to stop you there for a minute. So I've heard about them egressing, but I've never heard to say to recover. So,
yes.
So explain that to me a little bit more.
So if you signal through the B cell receptor and say to grow, and the B cell grows, it kind of ratchets down the B cell receptor a notch and says, okay, we're going to take this down. And then it grows again, takes it down further, and grows again, takes it down further. And it almost like it's trying to suppress itself, if you will.
Okay.
And when it finally gets to the point where it can no longer grow and proliferate, it no longer secretes those hormones that retain it within the lymph node, and then those CLL cells go out into the peripheral circulation.
So it's not responding to the chemical signals in that microenvironment
Yes.
where it was before to say, stay here, grow here, proliferate here, I'll protect you. It's saying, I'm tired,
I'm
let me
exhausted.
get out of here, and I'm going out into the periphery.
For the longest period of time, we thought that CLL cells were like these inert cells that didn't do anything because we were taking them out of the blood, studying them, and couldn't get them to do anything because they were exhausted. Kind of like me at the end of ASH.
All of us at the end of ASH.
All of us. And so they were exhausted, and what happens is they start getting their B cell receptor up and up and up and up and up. And finally, once it's up high enough to actually signal through the B cell receptor, goes right back into the lymph
antennas are
nodes.
up and they get the
Yeah.
signal, hey, this is where you belong.
Yeah.
You've got a microenvironment that's going to be very supportive of you.
And we can signal now.
Yeah.
So like the connection gets reasserted. So now, so if you do this kind of test called the gene expression profiling, it's a very common test we do
Right.
where we just, you gotta read out red, blue, on, off, and the difference between the cells in the circulation and in the lymph nodes and bone marrow couldn't be more different. Okay, so now that's like one key piece of the biology.
I'm going
Yeah.
to stop you there for a minute. So, do we have a sense, I know that the lymphocytes
Mhm.
reproduce in the nodes.
Mhm.
They're
And
also,
marrow.
and in the marrow. Do we have a sense of which is more predominant, or is it,
I don't think it matters.
varies? Doesn't
I
matter?
don't think it matters and we don't know.
All right, and it's also the spleen too. When
Mhm.
you're talking about nodes, you're
Yeah.
including the spleen and
It's a big
even
lymph
the
node.
liver to a lesser
To some
extent.
degree. Yeah. So the next thing, and this is really important for evolution of high-risk genetic markers, is that when somebody has CLL, from a variety of studies, it looks like about 1% of the CLL, somebody's entire CLL, burden of disease, about anywhere between 0.5 to 3, but on average about 1% of the cells are both born and die every day. So it's not like something that's just locked in place and you say, oh, I've had stable disease for the last 2 years, my counts haven't changed. Instead, it's a treadmill. And 1% is not a huge number, but 1% a day when you're talking about billions upon billions of cells actually is a lot of cells both born and dying.
And that's from the heavy water studies that
Yes.
we did. And that's also about the same percentage of red blood cells that live and die every day.
Yeah, about that.
Yeah.
You're right.
Yeah.
Actually, because a red cell lives about 120 days.
Yeah,
give and
roughly
take,
the same, yeah.
Not in you, but in me.
Yeah.
So about 1% per day. So now what we've shown, and Jan Berger presented this here, is if you give ibrutinib or idelalisib, well he did it with ibrutinib, but it's presumably true with both, you shut off that proliferation, and there's a little bit where the cells, now they can't signal in that microenvironment, right? So they can't be retained in the lymph node because it's the B cell receptor that keeps them trapped there. You shut off that signal and they go into the blood. Okay? And they're not happy in the blood. That's where they go to recharge, but if they don't get back into the marrow or the lymph nodes at some point, they start to die. So what we see is we're shifting that balance between being born and dying of these lymphocytes. That's why when somebody's on ibrutinib, their counts go up and then it takes forever to come back down. You're oftentimes losing like 1% of the cells per day. And it's this long, slow curve where your counts spike up for about a month or two as everything floods out of the lymph nodes and then slowly declines.
So I have to tell you that the analogy that I've used when I've talked with patients about CLL is I don't put up those complicated slides that show the BTK pathway
Yeah.
and where you block it. I show a B cell, and then I show a B cell with a condom on it. And so that B cell loses its ability to communicate and be in contact
And you're
with
saying we
anything.
do this, a
And
condom
that's-
blocks that. Okay.
Yeah, and it's sort of, but that's what these BTK blockers
Yes.
do. They kind of block it,
Yes.
and then it kind of dies of isolation
Yes.
eventually.
Loneliness.
Loneliness. It's like an incredibly oversimplistic, explanation,
But we'll
but
go with
it's
it.
a one step.
Yeah.
So let's take a break with that kind of
Imagery.
imagery, and we'll pick it up in a minute.
Sounds
All right,
good.
thanks.